Hi, my name is Peter Litsky. I'm a biology professor at the City University of Hong Kong and the co-founder of Singularis. And this is a research paper, and actually quite a good one. Research papers are still the main way scientific knowledge is communicated, stored, and reused. But with all due respect, if we are honest with ourselves as scientists or students, do we really read these papers from A to Z, from the beginning to the end, line by line? And are papers really the best format for communicating scientific knowledge? Research suggests the answer is no. Scientists rarely read papers linearly. Instead, they skim, skip, and navigate between sections to extract the information they need at a given moment. We call this strategic reading. So, if papers are a suboptimal medium for communicating knowledge, why do we still rely on them? We believe the reason is more institutional than practical. Academic incentives still revolve around publishing papers, so moving away from this format is difficult. At the same time, we believe the paper format contributes to many of the institutional problems we see in modern academia. People often talk about the need for reform, but there is still no clear alternative to the current system. Our view is that in the age of AI, the way we communicate research should look very different. And that is exactly what we are building at Singularis. We believe that representing research as an argumentation graph can be far more effective both for human understanding and for machine-assisted knowledge extraction. So let me show you what we have built so far. Right now, our platform works as a layer on top of existing research papers. Here, for example, we present the classic Watson and Crick paper in which they discovered DNA double helix and present the argumentation graph alongside this paper. You can see that the graph contains different types of elements connected by meaningful links. In the first row, you can see the facts that informed the research, Chargaff's Bayes ratio, pre-existing structural models. One was made by Pauling, one by Fraser, chemical considerations, and also contribution from Wilkins and Franklin that inspired the authors. Below, you can see Watson and Crick's analysis of existing structures and why they found them unsatisfactory. That analysis led to formulate a new research goal, to identify a better structural model. This, in turn, led to the main result, the double helix model of DNA. You can also see the analysis they applied to this model, the main conclusions they come up with, and a further hypothesis that nucleotide pairing could explain genetic copying and realization of genetic information. We believe that having such schemes alongside the papers can help readers understand scientific knowledge faster and more clearly. By clicking on the specific element, the reader can jump directly to the part of the paper from which that element was derived, and this provides context and also makes it easier to assess whether the annotation is accurate. At the moment, we are focused on developing a format that would be accurate, simple, and maximize the efficiency of strategic reading. In the future, we want to connect graphs derived from many individual papers to build argumentation graph for entire research fields. That could create a fundamentally new way for researchers to interact with knowledge. We also believe that other academic activities, such as peer review or even publishing itself, could eventually migrate into this system. We have many ideas about how this new format could help address some of the problems academia faces today. We would be very happy for you to try our platform. We need your help and feedback to develop an efficient graph format. If you can try it while reading the papers currently on your plate and tell us about the outcome, that would be amazing. Also, if you have any ideas, feedbacks, or interest in collaborating with us, we would love to hear back from you. Immortality and progress, stay tuned.